Hello Bruins, I hope you are enjoying your snow day, and I decided to make this screencast if you're doing the genetics problem homework, and if you're having trouble you can watch this. Now with the genetic problem set, first of all we're going to do is complete dominance, which is a simple monohybrid punnett square. Now what does that mean? It means that we're looking at one trait, and when we say complete dominance we mean one trait is stronger and will show up in its presence. So if the allele is there, you'll show that trait. And the other trait is a recessive allele or a weaker trait that does not show up in the presence of the stronger trait. So here, for example, cystic fibrosis is a disease that is recessive. So that means to not have the disease is the dominant trait, and to have the cystic fibrosis allele is recessive. So here, for example, if you look at this pedigree, here these two parents are shown, they're in blue because they are carriers. So whenever you're representing a dominant trait, we write a capital letter. So instead of using C for cystic fibrosis, I'm going to use a big D. So big D will represent not having the disease, and small d is having the cystic fibrosis. So if you have two parents that are carriers, that means they are heterozygous. So that means they have both alleles, and since cystic fibrosis is recessive, these two do not show the disease, but they carry the cystic fibrosis genes. So that means when they have children, it's possible for their children to have it. So this individual has cystic fibrosis, so she's homozygous, where these two are carriers, so they're heterozygous. Now what are the chances if both parents are heterozygous, and what's the percentage of their children to have that disease? This is what a punnett square does. So basically what you're doing is you're setting up the possible gametes. Say the father has, so you could put the father here or here, it doesn't matter. So the father's possible gametes is D, big D, and small d. The mother is big D, small d. So here if we put the father, so this row kind of represents meiosis, what would the possible gametes would be. So, and then the mother we'll put here, and then you bring it across and down. So this shows that the possible genotypes, so if the question asks you genotypes, that means what are the alleles. The possible alleles is being homozygous dominant, heterozygous, or recessive. So here in this case you have 1 for homozygous dominant, 2 for heterozygous, and 1 for the trait. Now if the question asks you for phenotypic ratio, that means the trait is either you are normal, don't have the disease, or you have cystic fibrosis. So in this case, you have 3 out of 1 of having the disease. So 3 would be normal and 1 would end up having cystic fibrosis. So in order for a recessive trait to show up, both parents have to be carriers. Okay. Now, what about an example of if there's a disease that's dominant? And for example, Huntington's disease is a dominant trait. So if you have the disease, then if you have the allele, then you have the disease. So we represent big H for Huntington's and small h for normal or not having the disease. So in this case, let's say these both parents are heterozygous, okay, and they both show the disease. So in this case, if we were to do a Punnett square, you would have 3 to 1 chance of having the disease. Okay, so this means this individual is probably homozygous, these two are heterozygous, and this individual is probably not affected. So now if say one of the parents had the disease and the other parents was normal, what would be the possible phenotypes? In this case, if I were to do a Punnett square, that means there's a 50-50 chance of the offspring having the disease, so 1-to-1 ratio. Now there's also something called let's see, this is the problem you have. So this is just giving you the scenario with the offspring, and you have to figure out what are the genotypes of the parents and the gametes and the offspring. So you're kind of working backwards. So a black guinea pig is crossed with the albino guinea pig, produces 12 black offspring. All right, so this is what you know so far. So you have a black guinea pig crosses with the albino. So these are the parents, and the F1 generation, they're all black. So right away that tells you the black is most probably the dominant trait. Then they tell you when the albino is crossed. When the albino is crossed with the second black one. So when the second black one of the F1 hybrids, so this is most probably a hybrid, because if this is a pure black and albino, then all the offspring must be hybrids. In this case, you have 7 blacks and 5 albinos. So if you look at it, it's about 1-to-1 ratio. So 5 into 5 is 1, and 5 into 7 is around 1. All right. So this is what you would expect if this is recessive. If we were to do a Punnett square, so this clearly shows a 1-to-1 ratio. Okay. So the question wants to know the genotypes of the parents. So the genotype of these parents must be pure black and recessive, because if it's pure black, then all the F1 generation would be black. So just to make sure, this is what you would had with the Punnett square, and this shows you would have all black. Now, just to make, and then albino and heterozygous, because you would take one of the parents, and this is what you would get. So the genotypes of the parents for the these guys are again big B, heterozygous. Gametes, possible gametes and offspring, is shown here in the Punnett squares. Okay, now besides complete dominance, you can have an incomplete dominance, and this is basically a blending effect, and you have an in-between trait. So when you see in this scenario, you'll always see at least 3 phenotypes. So in this case of flower, if you have a red flower, we have a pink flower, and you have a white flower. So if we had mated a red flower with a white flower, right? If it's purebred, so let's represent red as big R, and let's do white as big W, then all the F1 generation is going to be hybrid, and that's all pink. Now what would happen if you mate a pink with pink? Okay, so one parent would have a red gene and a white gene, a red gene and a white gene for the other parent, and you bring it all down and across, and the possible genotypes is RR, RW, WW. So we had one of those. Oops, I forgot to put that there. 2 that are heterozygous, 1 homozygous recessive. So you got 1-to-2-to-1 ratio in genotypes. Phenotypes, I have red, pink, and white. And that 2, I had 1 red, 2 pinks, and 1 white. Again, 1 to 2 to 1 ratio. So in incomplete dominance, your phenotypic ratio and genotypic ratio would be the same, 1 to 2 to 1 ratio. Codominance is where if you have the allele, you show the allele. So in this example, cattle have different fur colors. So a red bull is crossed with a white bull. So we'll use big R to big W again, right? So a red bull with a white cow has 8 offspring, so all the F1 generations are hybrid. And this is called roan. So that means if they're homozygous, they're red. If they're hybrid, they're called roan. And if they're white, they are called white. Okay. So, question says, what happens if roan? How would the rancher establish a herd of roan cattle? If you want all roan cattle, so then of course the parents, if you made a red with a white, all the offspring would be heterozygous. So if you had big R, big W, all the offspring would be roan. Alright, now blood types. Now blood types have multiple alleles, meaning there are several alleles that determine blood types, and it's also codominant. So if you have the allele, it'll show up. Now, if you are blood type A, the alleles that you would have, and sometimes they represent that with I and superscript, and for our purposes, we'll just use A for blood type A, or B and O for blood. So what happens is, on a red blood cell, you have carbohydrate groups. So say A blood would have a particular carbohydrates. So what happens, if you're blood type A, you're going to form an antibody against B. Okay, and let's say blood type B has carbohydrate groups of such structure, and blood type B will form antibodies against A. So that's why if you're blood type A, you can only get blood type A. If you're blood type B, you can only get blood type B. Or blood type O has no carbohydrate groups, so O can donate both to B and A. Okay, so if you're blood type A, your alleles could be big A, or AO. If you're blood type B, you could be BB or BO. And blood type O, you have to be OO. Now you can also be blood type where you have both the carbohydrate groups, and that's blood type AB, and AB also can get blood that is A, B, or O. Okay, so how do you predict? So say if the mother is blood type A and the father is blood type B, what can the offspring be? So if she's A, she could be AA or AO, and if he's B, he can be BB or BO. Okay, so let's set it up here. So let's put the father here. So if he's B, he could be either O or B, and if the mother is A, she could be either A or O. So if you were to do a punnett square, AB, AO, and if he's homozygous, then it would be AB, AB or BO, AO or BO. Okay, so the possible blood types for the offspring could be AB, BO, AO, but not OO. Well, none. That was A. Well, actually, she could be O, because if this was O, if she was O, and he was O, in that case, you could have OO. So this is how you would predict different types of blood types. Let's see if we do another one. Let's say if the parents are O and AA. She's homozygous. So AAOO, in this case, all the children must be blood type O. Okay. All right, sex-linked traits. Now, sex-linked trait means that to be a female, as you can remember, you're XX, and to be a male, you're XY. So what happens if a trait is on the X chromosome? So, for example, in color blindness, let's represent big B as normal and small b as colorblind. So if you're colorblind, the possible phenotypes is female normal, female colorblind, male normal, male colorblind. Now, if you're female normal, then the possible genotype is you could be homozygous normal, so this is how you would write it, or you could be a carrier since you have two X chromosomes. Now, for a female to be colorblind, she has to be homozygous. For a male to be normal, if he has that gene, he's gonna be normal. If he has a colorblind gene, because he only has one X, he's gonna be colorblind. So these are the possible phenotypes and genotypes. So let's have a scenario. So let's say the father is colorblind, okay, so and the mother is normal, homozygous normal. In this case, notice this one, you get 1X from the father and 1X from the mother. So this female is going to be a carrier, and the sons only get their X chromosome from their mothers. They never get it from their fathers. They get it from their mother. So if the mother has that colorblind gene, then the son can be colorblind. And then this female is also heterozygous. Now, if this mom, if we take this scenario where the mom is a carrier, and let's say the father is normal, then in this case 50% of your sons can be colorblind and 100% of the females will be normal. So males, that's why you'll see males more affected with sex-linked traits. Okay, so a calico cat, calico cats can only be females because they have two X chromosomes. So basically, if the female is homozygous with big B, she will be a black. And if she's heterozygous, she'll be calico. And if she is homozygous small b, she will be yellow. Males can only be black, and males can only be yellow. So what happens if you meet a calico cat with a yellow male? So we would write yellow male like this. Calico cat would be hybrid. And notice the mother gives that X chromosome to the son, and the mother gives one to the daughter and one from the father. So in this case, you will have one calico cat, which is of course a female, one male black cat, and one female yellow cat, and one yellow male. Okay, so you'll have 1-to-1 ratio. Remember that in this case, the phenotype to be a male and the trait. Okay. Now the last one, polygenic traits. Polygenic means that there are several genes that control a trait. So in case of eye color, there are 3 sets of genes. So what it's saying is here the mother is all recessive. So you have BGY, and she's all recessive, and the father is heterozygous. So if you had to do a Punnett square for this, it would be a 16-box Punnett square, and there's 8 possible combinations of the gametes. Now mother is all recessive, so she's always BGY. So we can kind of ignore the rest of the rows and just look at this. Now the possible combinations for the father, right, is, okay, so the possible genotypes for the father would be big B, big G, and big Y, all dominant. 2 dominant genes. And this is what you're given. Now, I'm not going to have you put these together, but what we're going to do is we're going to count how many dominant genes your offspring can have. So the number of dominant genes will determine the eye color. So for example, the mother is always going to give you recessive, but if in this case, a father will give you 3 dominant genes, in this case 2, in this case 2, 1, 2, 1, 1, 0. So if you look up in this chart, if they have 0 dominant genes, then your eyes are blue. And if it's green, you have at least 2 to 1 dominant gene. So this individual is going to have green eyes, green, 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 and this individual will have brown eyes. Okay. Now you can also do this mathematically. For example, if you say, what's the possibility of a child, say, being all heterozygous? Big B, small b, big G, small y, small g, big Y, big Y. So you could do this mathematically. So what you could do is say, okay, the possibility of having a small b is a half. Possibility of having, not half, it's when mother is 1, and the father is a half, so 1 times a half is a half chance to be heterozygous. And for mother is 1 out of 1 to be a small g, and it's a half a chance of getting a big G, so that'll be a half again. And the chance of you having a small y from the mother is 1, and a big Y from the father is half. So multiply all those 3, 1/2 times 1/2, that'll be 1/8. So you have 1/8 chance of having an offspring that'll be heterozygous. So I hope this helps. So come with questions in class, and we'll go from there. Well, thank you for watching and listening.